bioinformatics sequence search techniques to then gather the known enzymes from other plant genomes and go and search in our assembled genome. And when we do that, sure enough and encouragingly for us, we could find a number of hits. Number of hits for each of the enzymes that we know are in the biosynthesis of the secondary metabolites, and in this case we managed to do this for about a dozen of them. And then our bioinformatics really begins because sometimes when we do sequence searches, and simply because these computational algorithms are very sensitive, you might pick up a number of hits which don't necessarily mean that this is the enzyme that sits in that pathway. So to decipher this, we do what is called as phylogeny. Phylogeny is a way by which you sprinkle in some known members that you suspect your hit belongs to, and then you sprinkle in some distantly related members, so something which is little far away, and then you expect phylogeny to resolve and tell you that if your hit from the Tulsi genome is going sitting in the right cluster, that means it's a true hit. But on the other hand, if it goes and associates itself with the wrong subfamily, so that means it's an interesting gene, an interesting enzyme in plant Tulsi, but this is not what we are looking for, because it's not the enzyme that is there in this pathway, but maybe doing something else. Okay, so for this we also ensure that the functionally important residues are in place, and you can see all these which you see as OTE stands for Osimum tenuiflorum, which is the Tulsi plant. And these are all the hits that have responded. Isn't it beautiful that you choose a plant that you start to adopt? If you had the possibility to do sequencing, you can get the raw reads, you can assemble them using bioinformatic techniques, you can find the genes using bioinformatics tools, and then you can recognize domains, and then you'll be able to associate with the pathways of our interest. So that is the story which I want to say. And I don't want to just spend too much time on this because I have 3 other stories. And for me, the 3 other stories are shorter, not as much as the Tulsi, because Tulsi is always very close to our heart. So the second story, which in fact I had talked about, is nothing but Cissus quadrangulis, which is called as perendite in Tamil Nadu. And what does this plant look like? These are different pictures of the plant that we managed to grow in our own lab, and we also managed to take pictures of the plant blooming, fruiting, and so on. So this is a very beautiful experience. And what is of greater interest are the stems. And as the name suggests, you can see the four-fold symmetry you can see in the stem, and that's why the plant is also given the botanical name Cissus quadrangulis. And I understand in TNAU we have a beautiful rich resource of cissus. We have plants which are having 3-fold symmetry, not only 4-fold symmetry. So I'm sure you have more to say about this plant than I do, than our lab does. But then for us, from the medicinal viewpoint, what is interesting is it is very well known to be used in the case of bone health. Whether people go through bone fractures or joint problems, it has been used for many thousands of years as a medicine to cure fractures. In fact, it is so strongly felt that in Sanskrit, the perenda is called as harjor, meaning something which can bring in a broken bone together, right? So that is the power and proof of this particular plant. And not only that, it is known to also be an analgesic, treat infections, anabolic, promote weight loss, and so on. So what are all the secondary metabolites that confer this property? So that is our question. So the anabolic steroid, the one that is involved in fracture healing, people are still working to understand exactly and pin down the small molecule, but these are known to be estrogenic receptors. And in fact, this particular molecule called resveratrol has been implicated many times for both fracture healing as well as some other ailments. And thankfully for us, this particular plant is rather close to the fruit grape. You may not appreciate it by looking at the pictures, but these are also having tendrils, they are climbers and so on. So they have a lot of properties similar to grape. And the genomes of transcriptome studies results were available for grape, at least for the enzymes that are involved in the synthesis of resveratrol. So we definitely used, so that was our start point. And after we assembled the genome of Cissus quadrangulus perennial and got some statistics that we were satisfied with, we went and searched for these enzymes that are producing resveratrol, not only resveratrol, but we also went after other small molecules such as cetosterol, stigmasterol, and also it shares secondary metabolites as observed in many other plants such as quercetin, camphorol. Camphorol is also there in tulsi. Quercetin, as you will see, it is there more in other plants such as Moringa. Okay, so that is what takes us next to our third story, which is that of Moringa. Moringa is known to be a miracle tree. So tulsi is known as the queen of herbs, but Moringa is called as a miracle tree. So these are not given names just simply, but for reasons. And Moringa, for instance, every part of the plant has been implicated to cure one disease or the other, starting from the leaves until the roots. The leaves, as you know, it's used day in and day out to manage diabetes and many other ailments. But the roots, I believe, are used for cardiac ailments. So yeah, people would take some bit of the extracts from roots and use it properly with a proper recipe to cure cardiac ailments. Right. So what we had done in the past is to look closely at this Himalayan tree, Moringa oleifera, and of late, meaning for the past 3 to 4 years, one of my students, Shafi, has been closely comparing this with another species called Moringa concananis. As the name suggests, the Konkan should give you a clue that it is more observed in the Konkan regions, that namely the Western Ghats. And in terms of the geographical distribution, you can see whereas Moringa oleifera is known to be of Himalayan origin, Indian origin, it is spread all around. In fact, some of my friends who are in the US also say that they are growing Moringa. I feel very happy. So it is adapting itself to a variety of climatic conditions. And also Moringa is not only used to cure diseases such as to manage diabetes or cardiac ailments because they have wonderful metabolites, but they also are a rich source of minerals. And many may not know that Moringa leaves offer 30 times more iron than spinach. Can you tell me who in this audience are aware of this? Iron means we think of spinach. How many of you knew that Moringa has 30 times more iron than spinach? Those who know, please raise your hands. Some of my people are and some of my colleagues are, but the students, no, I also didn't know until we measured them. We measured them, and we measured also those of calcium, and it's supposed to have so much calcium, and it offers zinc and so many very nice minerals. And that is actually true for many medicinal plants too. But Moringa is well known to be a superfood. Superfood, and you can give it to people, children who are malnutrition. And that we know in our continent as well as in the African subcontinent, we have many children who are severely malnutrition. So the idea is you can grow Moringa and offer to such children. And that alludes us to the third property of Moringa, which is its strong drought tolerance. It can survive with very little or no water. Right? We cannot survive without water. Most of us is water. But Moringa is not only a miracle tree because it can provide secondary metabolites which can cure ailments. It's not only a superfood because it gives so many minerals, but it also is a very good model plant to show how it can survive in drought conditions. So anyway, so what we knew was this Moringa concananis had higher medicinal value, and this is somehow acquired very little attention. So Shafi, the student in our lab, started to compare these closely using transcriptome studies. So that's a story that I hope to very briefly talk to you about. So the leaves of the Moringa oleifera is shown on the left hand side. The leaves of the Moringa concananis is shown on the right hand side. So the pinnate conditions, they are pretty different. So it's not easy to even recognize that the second one, the Konkana one, which is also called as a wild Moringa, belongs to the same family. And look at its flowers, they are also rather different. The Moringa flowers are pinkish, whereas the Moringa oleifera flowers are pinkish, but the Konkana one are more white, and they have slightly different structures. So this is to show you that the morphology of the two trees are different, but what we really wanted to pursue is how are the distribution of the secondary metabolites different from each other. Can we actually show using in vitro assays that the two plants have slightly different properties, such as we can use some in vitro assays to show that it is capable of weight management, right? So we can go to that. And like I said before, it's a miracle tree because almost every tissue of the plant is used for one medicinal cure or the other. And we also know, because of our experience in oleifera, that different tissues of the tree hold more of certain secondary metabolites. And I had mentioned this in my previous talks, so I don't want to repeat it. Quercetin and camphorol are more in the flowers, and then you can see benzylamine is more in the pods, and so on. And this is now surrounded by all these secondary metabolites of our interest. You can see urusolic acid is there, but it is less. And quercetin on the other hand is more strongly associated with this particular tree, medicinal tree. And then we did the same, we repeated this for the different tissues of the wild Moringa, the Konkana one. And we also could, Shafi could assemble the two transcriptomes using the genome that was published by the Chinese group, and then these values, the statistics values are shown as a table for the two plants. Okay, armed by these two transcriptome assemblies, we then decided to concentrate on only 3 metabolites, like how starting from 43 metabolites we concentrated on 12 of them for Tulsi. To compare the two types of Moringa, we decided to concentrate on only 2 secondary metabolites and the enzymes that are involved in the synthesis. Quercetin, and there are as many as 6 different in 1, 2, 3, 4, 5, 6 different enzymes that are involved in the catalysis of the initial product, the primary product, going from one to the other, acquiring its chiral centers and so on. Finally, it gives rise to the quercetin, right? And then on the other hand, benzylamine. The molecule looks so simple, but it is very powerful, and it is found in abundant conditions in this particular plant. It has another name called Moringa. So you can imagine how much this benzylamine is associated with this particular tree. And we also decided to focus on a third secondary metabolite called chlorogenic acid, for which as many as 9 different enzymes are involved. And in particular, we focused on HQT, HCT, because these are very similar enzymes. They are more than 30% identical. And at some stage, if the HCT is more, it can give rise to a certain secondary metabolite. If the HQT is more, it can give rise to another metabolite. So you can imagine how much the presence of these enzymes will contribute to what sort of secondary metabolites will come. So what does this heat map say? The heat map is no longer concentrating on the medicinal value, the secondary metabolites and so on, but instead it is showing us the relative abundance of the enzymes that are back in the synthesis of the 3 secondary metabolites that I said, quercetin, benzylamine and chlorogenic acid. And since there are 8, 2 and 9 different enzymes involved, this heat map is a way for us to compare and appreciate how not only the two plants differ from each other in harboring the enzymes of interest, but also how the different tissues, namely flower, leaf, stem, root and so on, are holding the different enzymes. These have been carefully calculated, and these are called as TPM value, total parts per million of the RNA reads that are observed in the different tissues between the two plants. So whichever is dark green means it's more abundant. The lighter the green color, the lesser the amount is. As you can see, the last 3 enzymes, this the second, third and fourth enzymes in the quercetin pathway, it's much more abundant in the stem of the wild Moringa. So this we think is kind of a minor breakthrough. It is an encouragement for us to believe that this may be some of the reasons why the wild Moringa has better medicinal properties, because it has more of the enzymes that are required for that secondary metabolite quercetin. And quercetin is well known for its anti-diabetic properties. And what I am not able to go through the details, but for all of these enzymes we have carried out what are called as QTR, PCR, by which RT-PCR experiments by which we can quantify and validate that indeed these enzymes are observed in large amounts in the tissues of the interest. And you can see that these values are corroborating very well with the RNA and the transcriptome assembly that has been used to compare the two plants across the tissues and so on. Okay, I think I will just conclude the story by telling you that we have indeed carried out not only the quantification of the metabolites, and you can see what is shown in dark purple color is the wild Moringa. And you can see that chlorogenic acid, for instance, the amount is as much as 244 in the wild Moringa, and it's much less in the Moringa oleifera, where you see only 171. Shafi has also carried out in vitro assays that directly impinges on a better understanding of the lipid management and how it reduces obesity in humans and so on. But almost all this comparative analysis I have talked about are unpublished. We are preparing the manuscript, so hopefully in my next visit I can share some of these details with you when hopefully we would have published. Okay, so that then takes us to the last story, and this is also the one which is the most recent. It is the story of Sankakushvi. Sankakushvi is again not only an ornamental plant, it is a plant that is used in religious situations, and it is a very holy plant, and the botanical name is Clitoria ternitia, and it is been used again for thousands of years for several medicinal reasons. It has been known to alleviate microbial infections, act as an insecticide, and it is cytotoxic, anti-inflammatory, anti-cancer. And this picture kind of emphasizes the original and the most prominent reason why the Sankakushvi flower tea is used by humans, especially in some tribal populations. People still use them when a lady is going through childbirth. The childbirth is associated with huge amount of uterine contractions and pain, so it is believed and documented that consumption of this tea reduces the pain and also eases out the childbirth. And back end, what we know from the scientific angle is that the plants produce this sort of special secondary metabolites, which are also called as cyclophytes. There's something very, very special about them. They are not small molecules like the quercetins and the benzylamines and the chlorogenic acids, uracilic acid which we saw before. These are themselves proteins. And these are also held together by covalent cross-links called disulfide bonds, and every time you see a C, that means in the primary structure is a cysteine, and that means this C and that C are committed into a covalent cross-link, a disulfide bond. And you can see that there are lots of these cross-links going on, like a metro map. So that means there are many disulfide bonds. And every time there is a disulfide bond, the covalent cross-link, the protein is said to become stronger and stronger. Stronger against what? Against thermal stability. Because proteins have a tendency to denature, right? If you boil the milk and then you overdo everything, then it can get denatured. But these cyclophytes, on the other hand, they are known to be stable even at 120 degrees centigrade. And this is possible because it has 3 disulfide cross-links and also it cyclizes. Cyclization also gives higher thermal stability. So the N and the C terminus are forming a peptide bond. So this is the beauty of this cyclophyte, and there are several structures known, not of the cyclophytes in Sankakushvi so much, but from other plants. And this is the structures of cyclophyte from the related plant. And what are all the plants that produce secrete cyclophytes simply for, they do it for their chemical defense, right? And that's how we started the discussion, isn't it? The secondary metabolites are produced by plants because they cannot run away from pests or herbivores. Okay, so all of these plants that are shown here are examples where they naturally produce cyclophytes, and one of them is, you will be interested to know, is nothing but coffee. Coffee, and in fact one of the species, I am sure some of you can relate, that you have a coffee variety called Robusta. Robusta because it's robust against pests and herbivores, and because the Robusta coffee produces more amount of this cyclophytes. Right, anyway, the structures are known from one of these. I will not go into too much detail except to tell you that a good amount of structural information are available from all these plants, and for each of them the secondary metabolite of interest are the cyclophytes. And like some of the conus toxins, the cyclophytes not only are proteins but they have genes behind them. These are not like the small molecules which depend on enzymes to get them synthesized by the plant, but there are genes encoded. There is a pre-pro-cyclophyte which gets cleaved and cleaved again until it is ready to produce its and kind of present itself as a primary defense for the plant. Okay, so that is the other fascination. So where are we doing with respect to our transcriptome analysis? That's what I want to tell you. One of my collaborator student called Neha decided to focus on the Sankakushvi and extracted the cyclophytes. Why the extraction? Because first of all, these are proteins that behave as secondary metabolites, and also these are thermally highly stable. So she would just boil the plant, the leaves and flowers and all, and then go after the traditional extraction procedure, which gives a large amount of cyclophyte, which she then used it for transcriptome sequencing. And in fact, she took other tissues as well, flower, leaf, or stem, and then she would take the RNA and try to purify it and take it forward, and she took it forward for transcriptome assembly, much like what was done for Moringa. Right? And here I had very expert collaborators, Dr. Radhika Venkatesan, who was in NCBS like a year ago, and she had expertise not only in handling plants and extracting metabolites from them, but she was also particularly interested in plant-insect interactions. And then I also was fortunate to have another collaborator who is none other than my PhD thesis supervisor, Professor Balram. And as you know, you may know, Professor Balram is a chemist, and he is very interested in small molecules and especially structure determination using mass spectrometry techniques, and that's where he contributed. And together we started asking questions such as how many cyclophytes are there in this plant of interest, and we found that we pick up all the known ones and also we pick up some unknown ones. Some normal cyclophytes which we see only in this plant, and that's understandable because if the plant is very effective herbivore, it must be producing a large number of cyclophytes, different types of cyclophytes, and that is shown here as some of them which are actually novel and present only in this particular plant and so on. This also shows you the pre-pro arrangement of the genes of interest. So that is about the cyclophytes, but we didn't stop there. Whereas we don't have enzymes which produce the cyclophytes because they are genes themselves, we asked, because these are disulfide rich systems, what about the allied enzymes which are known to help, such as prolyl oligopeptidases, thioredoxins, and also the fact that it is a cyclic molecule, that means N and the C-terminal amino acids form a peptide bond. So there is a particular enzyme called aminopeptidase which is used as a catalyst. So we went after these questions, and we found, for instance, we can evidence novel cyclophytes Using mass spectrometry, we were able to attribute its sequence and enlist them, and there were more than close to 100 cyclophytes in this beautiful plant. And apart from the cyclophytes, like I said, we concentrated on enzymes that facilitate disulfide bond formation, such as protein disulfide isomerases, which are used as catalysts in different parts of the cell when it is the disulfides are vitally required to form and give them thermal stability to the protein of interest. And we found that several PDI, as it is called in short, in fact are endemic to this plant. And we see that a number are co-clustering with the known PDIs from other plants, but one or two cases are shown in these ellipse here are observed only in Sankakushvi, suggesting that there must be a very specific requirement and recruitment process by which different PDIs are simultaneously expressed and recruited in different parts and tissues of the plant of interest. And likewise, we also asked the question whether we are able to find the enzyme that is used for the cyclization event, because the cyclotide is cyclic, right? And this is commercially very important in the industry, because many people would like to cyclize peptide of many types for different reasons, and they always find it very hard to know which is the enzyme that will do it. So when we presented it in different places, people were more interested in this enzyme than the cyclotides themselves. So anyway, we were grateful to find a few entries of this particular enzyme, and in fact we found multiple copies of the many transcripts of this enzyme in this plant, and these are also shown as a circle. And like I said in the beginning, and we will allude to it in the workshop later on, only if it co-clusters with the accepted other enzymes coming from other plants do we accept them as a true hit. And in fact these hits co-cluster very well with other known asparagine endopeptidases. And therefore we are now convinced that these are perhaps the true enzymes required for that reason. Okay, so with this I think I will really stop this fourth story on the Sankakushvi. Both our stories on Tulsi and Sankakushvi acquired a lot of attention because it reaches a community, right? So we went on press, and especially for Tulsi we had a very pleasant experience, because I found myself getting interviewed in different languages, first in Hindi, next in Tamil of course, and then Kannada, and then finally in English. So our team had a lot of fun doing all this, but what I want to do now is to tell you who are the people involved, my acknowledgements. Like I said, we started off with Tulsi, and when I said shall we work on plant genomics, my entire lab raised their hand for volunteering for this new work. So we are all there, plus none of this would have been possible without the very nice lending hand from Professor Ramaswamy, and also the CCAM colleagues, Dr. Malali Gowda, who was himself a plant genomics expert. And this was these are the people involved in Tulsi, in Perendai. We had our lab. Okay, this is now capturing ourselves in action. Like I said, we used to get together and sit and discuss almost on a daily basis. You can see several people sitting here and discussing. Here is a bike. Somewhere Meenakshi is also there, I'm sure. Yeah, so we can see that we are all sitting and discussing and trying to understand all this. Okay. Whereas in the Moringa, we had reduced to a small set of active people, but all of them had actually handled the raw data. And you can see definitely Meenakshi is here and Advait is there. And Shafi, who is a person who is looking at this cross comparisons of two types of Moringa, the wild one, is here. He is the one who is very quiet but very efficient, because he not only did the transcriptome, he would run around, go to Transdisciplinary University, which is an Ayurvedic, which has an Ayurvedic institute, and carry out the wet lab assays as well. And he will come online for the workshop. And we also had for the Sankakushvi a different team, like I said, Neha, who is a very, very effective student, who did all of these work, the transcriptome work, the mass spectrometry work, and the behavioral studies in worms, which I have not talked about. And plus, she also updated our disulfide database. She was very efficient. She managed to shuttle between 3 labs and kept smiling. And these are some of us who kept troubling her, I suppose. But no, I was very fortunate to have collaborated with Radhika and Professor Balram on this project. And last but not the least, I would like to stop by saying giving my acknowledgements to none other than my mother. My mother, because she was a person who used to give away tulsi seeds as a gift to people, and how much of foresight she had. And she had actually enthused me to think about plant as medicinal units. And also she was the one who told me that there is some other species of Moringa called white Moringa, which you have to go after because they have conferred better medicinal potential. And thank you to my mom for including me. If you have any questions, I am happy to take at this point. So these next 2 slides will provide you some glimpse about what the workshop is coming about, and Dr. Abhay Joshi has prepared these 2 slides along with some introduction on what is we would like to know from the bioinformatics sides on the genomics and transcriptomics. And I hope it will also help you to get prepared and prime into learning a lot more about the bioinformatics aspects of doing computational genomics.
To look for the bioavailability of those nutrients or the metabolites, because we are applying on the wounded person. So instead of swallowing it, we apply it on it. So will it be okay to look at the bioavailability perspective?
Yeah, I think if plants provide them, human body is not able to absorb all of it. And that is true, that may be well known for turmeric and the curcumin that the plant offers. But even here, the bioavailability is an important question. And in fact, we could add it along with other, time and again it has been proven that if you mix it with certain other compounds, the bioavailability increases. In this particular case, it may be a different one, but in general, people notice that pepper and honey provide better bioavailability for the secondary metabolites. So instead of focusing on the enzymes and the commercial aspects about being able to synthesize them in large amounts and so on, what one can also do is to provide better application in terms of providing better protocols of using these plants. And one of them is bioavailability. And then another is convenience of applications. That could be, because some of these ideas could be patented. And for instance, if you have strips by which you can just put as a band, along with a band-aid, can this stay on and provide the better healing properties. We could also have some surface entropy increase so that the important molecules are held and provided to the tissues in better efficiency. So these are some of the open questions for which we need innovative ideas, and maybe some of the young minds will come up with them.
To present a vote of thanks. On the outset, I'd like to thank the Vice Chancellor who has proposed this endowment lecture, and the Dean School of Postgraduate Studies who have sponsored this endowment lecture. And we also thank the Registrar for his special address on this occasion. We thank the Director CPMB who has been instrumental, the former Director CPMB who has been instrumental in composing this MOU, and then the present Director who has been instrumental in guiding us through this type of programs. And I would like to thank the guest speaker, Dr. Sowdhamini Madam, and her lecture has shown how passionate she is about the plants and the work that she is doing. It's really inspiring. I'm sure all the young minds will take inspiration, and then could have kindled their interest to work on such aspects. And I thank all the student friends for their patient learning, and I'm sure you make the most of it and have more collaborations with Madam. And this program is to continue with the workshop. I hope you all get benefited from the workshop also. And thank you one and all.